it will produces fluorescence coloration. That's why it is called the chromogenic substrate method. The second method of determining fecal E. coli or fecal coliforms is called the chromogenic substrate, because it produces fluorescence condition in the test tube when it is hydrolyzed. So that's why you can determine whether we have a total coliform, or more especially what we have a fecal coliform or not. So now let's talk about some sets of confirmative states, which is going to tell us whether we have a confirm to make us confirm that whether we are having fecal coliforms or not. Because remember, false positive will be really, really bad, and false negative will be much more bad than that. Because that's why you need to go through very, very steady steps, very many reactions to finally confirm whether we have any water contamination with fecal coliforms or not. And all these reactions I am going to talk about now is account with different genera of enterobacteria. As you have known, the tests are called IMViC test. So this is, you remember always the name of this test. It is called the IMViC test. Okay. Or sometimes this I is written in short form. Why? Because I means indole, M means methyl red, V means Vogt-Prosker, and C means Simon's citrate. So we have indole, methyl red, Vogt-Prosker, citrate test. I is to actually make it sound familiar for us, IMViC. Otherwise IMVC it's tough. IMViC is really, oh yeah, that's why. Okay. Now what we have to perform this test, we have different tryptone broth. We need a tryptone broth. Okay. So we inoculate this tryptone broth with our samples. And why we are doing these steps? In all the steps I'm going to talk about, we are doing these steps to actually distinguish between differentiate between the species of enterobacteria. So whether we have a E. coli or we have a Klebsiella or Enterococcus genera, we need to determine that, because it's important to determine that whether we have a fecal coliform or not. Normally what is the funda between them we're going to discuss now, but we use different kinds of reagents. So now in all these reactions what we are doing, we are using reagents. In the previous reaction we have talked about ONPG and MUG for these enzymatic reactions. Now in this case too we are using reagents. Why we are using reagents? Because in this kind of reactions what is happening, reaction is taking places because we are actually using the property of bacterial cell to ferment something, or to digest something, to hydrolyze something, and to make a product. So how can we determine whether we have a product out of something? Suppose we have a medium, and we have a medium which is for example it is made up with tryptone. For example in this case we are talking about tryptone, so we have tryptone. Okay, and it is this tryptone is hydrolyzed or utilized by some sets of bacteria. For example, some of them can hydrolyze this bacteria, can use this amino acid to produce their products, and somebody uses some of those bacteria use them, and finally they hydrolyze, and we have a hydrolyzed form of this tryptone, So how can we know that this tryptone is hydrolyzed? Otherwise any visible changes occur, because in this kind of reactions no coloration, natural coloration is not occurring. So how can we determine whether our tryptone broth is hydrolyzed or not, or is taken up by bacteria for their purposes or not? So to do that we use some reagent. That reagent is going to react with the product, or the product of the reaction, finally give us a coloration most of the time. Because if we see the coloration, we can determine yes, the reaction is taking place, and after doing that we can confirm that our substrate is hydrolyzed to this hydrolyzed form, because only the coloration can be took place when the hydrolyzed form react with the reagent. That's why we have to add reagents in all these reactions. We'll go on and talk about. We have to add reagents, and by adding reagents we are actually improving the chance of seeing the coloration. So if we have to look look from the outside what is happening, then we need to add these reagents. That's why they are important. So in this case we are using reagents, we call them the COVAX reagent. In this reagent we have several different compositions. I don't recommend to remember all the composition, but remember that COVAX reagent is added. It is actually made up with hydrochloric acid and another chemical which is called dimethylaminobenzaldehyde and amyl alcohol. So this reagent, this dimethylaminobenzaldehyde is a reagent which acts well in the presence of acid. That's why you give hydrochloric acid, and when it reacts with the hydrolyzed form of tryptone, then it will convert it into a yellow colored compound, and we can determine whether the production of yellow colored compound is done or not. From this we can determine whether we have a fecal coliform like Escherichia coli, or we have Klebsiella genera. So in this case, this is the reaction step. So we have a normal uninoculated tube, which is called the normal broth or the simple broth, and the broth is inoculated. After the inoculation of the broth and all this thing is done, we add inoculation, 24 hours of incubation. After doing that, all the tryptone has been hydrolyzed. If the bacteria are able to hydrolyze, then we add this thing, which is called the Kovacs reagent. The Kovacs reagent coloration is like this straw color. After adding it, if the colors remains like as it is, we haven't produced any hydrolyzed form of tryptone. But after that, if we produces a coloration of this, coloration of a pink, like the coloration of red, not actually pink, it's coloration of red, then you can finally say that yes, the tryptone is hydrolyzed. And when the tryptone is hydrolyzed, tryptone is broken down, it produces a byproduct. The byproduct is, in this case, is indole. So this is the overall IMViC test. In IMViC test, we have indole test for the first place. So what is going on in this case? We have tryptophan amino acids. It can be broken down by the enzyme tryptophanase. So the bacteria who have this tryptophanase enzyme, which can only be able to break down this tryptone amino acids. So suppose some of them have this tryptophanase. So they secrete this enzyme, and they'll break down this tryptophan amino acid into indole and pyruvic acid, and it will produce ammonia. This is important because the indole will react with the former different chemicals like Kovacs reagent. Here we have Kovacs reagent. So we have the Kovacs reagent. So it will interact with Kovacs reagent, and finally it will form this red color form. So in this case, this is the red color formation. So red color layer in the upper side of the organic sample. Okay. So from this we can determine whether we have produces something or not. Okay, so that's how we can determine between them. So if we look at the previous example like this, so some of those bacteria can utilize this, and some of them cannot. That's why you use this selective process to go through, and all the process we will discuss about will be the selective process, and the selective processes will help us to determine whether we are talking about fecal coliforms like E. coli, or we are talking about the enterobacteria, other types of enterobacteria like Klebsiella and all this. Okay. So now let's talk about methyl. So the question can be arised that the first reaction we determine whether it is E. coli or Klebsiella species like that. So how can we go, why we are going into another steps of reactions? Because many times this type of reaction can give us a false positive, can give us a false negative, and that will make the trouble for us. That's why we need to be more and more confirmed. So that's why let's do some sets of experiments more. For example, the second set of experiment is called the methyl red Voges-Proskar experiment, or MRVP test. And MRVP is combined together because it accounts for the same type of medium, because in all those cases we need a glucose broth, the broth where you have a glucose for a main carbon source. So bacteria will be very happy because they have the glucose carbon source, and they use glucose as their earliest preference as a food source, that they can take the glucose. So inoculate all those glucose broth with a water sample. Then after 48 hours of incubation, we add those drops of MR. MR means the methyl red. That's why it's called the methyl red. And Voges-Proskar, in the case of Voges-Proskar, we have another sets of reagent, which is called the VP reagents to the tube. So as we know, what are the function of the reagents in all these cases. If in this case what we are actually test is based on, both tests are used to help differentiate species of family of enterobacteria. The same sets of tests which is using in case of in the previous test indole is again for the same purpose we are doing this test too. So the MR test for acid end products, and the VP test is for acetoin end product. So what do you mean by that? Because to break down this glucose into smaller particles to finally derive energy from it, we have several different processes to go on. In the bacterial cell, they have major two processes to go on, two pathway to produce all those energy steps. One is by this acid end product production, and one is by the acetoin end product production. So you have go through the acid end product production. Those are some of the different genera of bacteria, and the acetoin production can be done by another set of bacteria among the Enterobacteriaceae genera. Okay, so we use glucose broth. It is very, very simple. We use methyl red and the Voges-Proskar reagent. The Voges-Proskar reagent, what we have this potassium hydroxide we have alpha naphthol and ethanol as the reagent purposes. Don't want to remember this because it will be really puzzling for you. So what is the main funda of that? Again, MV, methyl red and Voges-Proskauer. What the name is, because we are using methyl red indicator and we are using Voges-Proskauer reagent. In the methyl red indicator, what we know about methyl red indicator in our basic chemistry classes, that methyl red is an indicator for acid production. So if acids are produced via different reactions, then the methyl red will turn red in color. So what is the principle? We have glucose, and when you have the glucose in the main carbon source, because we add these water samples in a glucose medium, then what we can do, we have two pathways to utilize this glucose, and this pathways is for bacteria. And we have acidic pathway, and another pathway is the neutral pathway. If we talk about the acidic pathway, it will produce the mixed acids, and after production of this mixed acids, the pH will really fall up to 4.4 or less than 4.4. When it falls less than 4.4, that will eventually gonna turn the methyl red's color into red color, and when it turns the color of methyl red into red, then it will be indicated. We can see the coloration, and we can find there this MR positive test. And most of the time, the Escherichia coli, not most of the time, all the time, Escherichia coli have an MR positive test. Okay. So they always they always want to go through the acidic pathway, and they produces this red coloration by the attachment or the reaction with the methyl red, and turn the color of methyl red into red color. That's why I call it the positive for MR. So all these fecal coliforms like Escherichia coli will be positive for MR test. Now let's go to the VP test. So if it is go through the neutral pathway, then we can test it with the help of VP of Voges-Proskauer test. So now let's talk about it. So the neutral pathway, which is neutral intermediate, will be produced if it will produce the acetyl methyl carbinol or acetone. As we know, we have talked about this acetone intermediate. It's called the acetyl methyl carbinol production or acetone instead of acidic production. So that will react with Barrett's 1 and Barrett's 2 reagent, which is a part of Voges-Proskauer reagent. Voges-Proskauer is made up of two types of reagent. One is Barrett's 1, another one is Barrett's 2. So these Barrett's 2 types of reagent, Barrett's A and Barrett's B, will react after one by one. After addition of these reagents, it will finally form a pink coloration. So when we form the pink coloration, then we can say that we are going to positive Voges-Proskauer VP, and that's for Klebsiella species. Okay, so this is another sets of reactions. So that's how the reactions are going on all the time in our place. So what we have to do, we have to have a glucose broth, and we'll differentiate this glucose broth in two different test tubes, and after making it to divide into two different test tubes, we are adding after inoculation in these two test tubes for 48 hours, we add this MR into, or methyl red into one tube, and we will add VP, that means the Barrett's 1 and Barrett's 2 into another, and after adding all those things, we look for the result. If we have a red coloration in after addition of this MR, we finally confirm that we have a MR positive. If we do not have any red color, we call it MR negative. And in case of Klebsiella, if we test for a Klebsiella with this MR test, it will be MR negative. If we test for Escherichia coli, it is positive, as we know. And in case of VP test or Voges-Proskauer test, in case of Klebsiella we have positive, but in case of Escherichia we have negative. Okay, so that's how it works actually. And now let's talk about the result. If we're talking about the result, what will be that? So in methyl red test, the result is that, so it's a normal broth after addition of methyl red's color is yellow normally. That's why when we add to the methyl red, for example, all those bacteria which cannot go through the acidic steps, it will remain neutral. That's why the color of methyl red remains yellow in those cases. But in other test tube where we have fecal coliforms in it, like Escherichia coli, they will produce acidic components, and that's why the methyl red color changes into red color here. So this contains Escherichia coli, you can say, and this contains Klebsiella, for example. Okay, so that's what happens. Now let's so red is positive for MR and yellow is negative, which is Klebsiella. Now let's talk about the Voges-Proskauer VP test. In the VP test, what we have, if we add after addition of Barrett's 1 and Barrett's 2 reagent, we can have a negative which have no coloration, you have positive you have pink coloration, but you cannot determine between pink, because in these cases after addition of VPF Barrett's reagent have a slightly slightly blackish yellow color. That's why the color with the pink, it will turn a slightly brownish color in this case. So you can actually say this is a pink. Is this a pink? No, this is a pink coloration, because in pure condition it's a pink coloration. So after having this pink coloration, so if we have a pink which have positive presence of Klebsiella is confirmed, and if you have no pink coloration, then you have negative and presence of Escherichia coli. So in this case, there will be Escherichia coli and there will be Klebsiella. So that's how we can determine it. Now let's go to the last sets of our IMViC test reaction, which is C for citrate test. So as the name suggests, again in this case we are using citrate slant. So the slants mean the slant is actually meant by we are, and now in all those reactions we have talked about broth, which is a liquid culture medium. A slant is a solid culture medium. By addition of agar, we convert those broth into a solid state, and we used to make is to land our test tubes. So it's a very bad drawing of test tube, but let me convert it into, yeah, so it can be a slant. So now it looks like a slant little bit. So this is a slant, for example. So what we have this agar with the medium, so it's a solid, and we have a test tube like that. So this is called a slant. I can say this is called a slant. Okay, so what we can do, we can make the slants out of this citrate medium. So use this citrate medium. Okay, and not only with citrate medium, but we also in the citrate medium, what is called a Simon citrate, as per the as discoverer of this or the inventor of this medium. We have the agar containing sodium citrate, which is the main carbon source for the medium. So only those bacteria will lead to positive test for these reactions, which we can break down or can take sodium citrate as their main carbon source, and the production of their byproducts will react with the sodium citrate and ammonium ion which is present there, and it will form a different coloration, as you know, and it is also have a pH indicator, bromothymol blue. So what does it mean? If you have a pH indicator, bromothymol blue will turn blue color and green color, or between blue and green color in all the steps. Okay, so if it produces any basic region, then the coloration will be blue. If it is in normal state or acidic condition, the coloration will be green. So normally the Simon citrate is normal times, in the formal times it will produce the acidic content slightly. For 6 to 7, the color of the Simon citrate agar remains green, which is the normal color of the Simon citrate agar. So if there is any bacteria present there, we can use this sodium citrate as the sole carbon source. They can use this, and they can produce their byproducts, which will give rise the pH, and as a result of it we have a slightly positive pH, and that's why the bromothymol blue will change color from green to blue, as we have talked about. Okay. So if I look at the principle of the citrate utilization test, we have Simon citrate, which is green in color, which is here as a slanted situation. So we have two slants, same types of two slants, and we use two types of medium in the slants. If we can do that, it acts as a whole carbon source. Then Simon citrate media have citrate, and the citrates can be broken down into pyruvate as throughout the different steps of reactions, and this pyruvate is further broken down into carbon dioxide, sodium, and water. And so the normal process of normal metabolic pathways, it will make pyruvate from this citrate, and when it made pyruvate, it produces carbon dioxide and water as byproducts, and also sodium, some amount of sodium. And when this this sodium, carbon dioxide, and water reacts together, it will form a compound sodium carbonate, and the sodium carbonate is a basic. So as the sodium carbonate is basic in nature, it will make the pH alkaline. It will rise the pH for a while, and when it is rise the pH, as we know, if we have rising of pH, the Simon citrate medium it contains bromothymol blue. The bromothymol blue turn its color from green to blue, and result of it will turn the blue coloration compound. That's why we call it a positive test. So if we have some of the bacteria, we can utilize it, then we can have this blue coloration. Okay, so positive taste in case of Klebsiella, Enterobacter, and Citrobacter, and that's why this Klebsiella genera, all this thing, they can utilize this, and they go through the acidic. They are not going through the acidic steps. They are going through the neutral media, and finally the basic steps to do that. And finally they produces this sodium carbonate via their normal metabolic pathways, and they turn the color from green to blue. And we have a negative taste for Escherichia coli because they do not produce all this upstream. They do not utilize citrate as sole carbon source. They have to utilize it whether either glucose or lactose. They can also use lactose. In case of indole taste, this Escherichia coli will give a positive taste, and in case of in this Klebsiella will give, in those in case of lactose fermentation, they will give a negative taste. So Escherichia coli can ferment lactose, can ferment citrate, and they go through the acidic steps. That's why they change the color of methyl red. But the Klebsiella utilize citrate, not they cannot utilize those lactose, and they go through the neutral step and finally produce an alkaline intermediate like sodium carbonate. And what is the result in this case of citrate utilization step? If you are looking at it, examine for growth, for example, these are the slants. If we have green color slants, the color is really great in this green coloration, and in this case, so when the color changes into blue, we have this blue coloration. In case of those Klebsiella genera, they will turn the color blue. So all these sets of reactions we have done, we have to determine, to distinguish between whether we have fecal coliform or the Klebsiella genera. We have to determine between the Enterobacter genera. And finally have this is the positive state test for Klebsiella Enterobacter, and we have a negative test for Escherichia coli, for example, in this case. Okay. So this is the different sets of steps of reactions that we can go through to determine finally what we end up with, or we have a sample, and whether we have a sample of Enterobacter genera, whether we have a sample of fecal coliform or not.